Good afternoon. It's my pleasure to welcome you all today to our Big Data Symposium, where we'll be looking at how you can utilize big data to tackle infectious disease and antimicrobial resistance. This is the final event of a series of symposia showcasing the Bloomsbury Sets projects. Today, we have 4 of our projects being presented, and we're delighted to have an external speaker joining us from the Infectious Disease Data Observatory. The session will be recorded and will be posted on the Bloomsbury Set website after the event. I would ask for anyone that's listening to today's presentations live to put your questions in the chat box. Following the initial presentations, we will then be having an open question and answer session. So please do engage to make that session as interactive and as interesting as possible. So before we move on to the talks about our research, I'm going to give you a brief introduction to what the Bloomsbury Set is. So the Bloomsbury Set programme is hosting today's event. This is a programme of knowledge exchange funded by Research England as part of the Connecting Capabilities Fund. So if we can move on to my next slide, the focus of the Connecting Capabilities Fund really is about collaboration. It was established by Research England back in 2017, with the first programmes coming online in 2018. We've had 3 years funding to bring us up to date, but the Bloomsbury Set really is part of this collaboration, joint working between universities, business, and external partners to ensure the best knowledge exchange, practice, and activities in the field that you're working on. The Bloomsbury Set is an interdisciplinary programme which incorporates 4 colleges which are part of the University of London. So the Royal Veterinary College, the London School of Hygiene and Tropical Medicine, the London School of Economics, and SOAS University of London. And these core partners in the programme work together with LIDC and external partners from industry and other stakeholder sectors to look at these important questions in infectious disease and antimicrobial resistance. If we can move on to then look at what the Bloomsbury Set actually does, this is an interdisciplinary programme. So from those 4 partner colleges, you can see quite a range of expertise, both in STEM and SHAPE subjects. And the idea of the Bloomsbury Set is bringing those together under this knowledge exchange umbrella to really support a One Health approach to deliver both human and animal health benefits. When we talk about One Health, what we mean is bringing that human, animal, and environment sectors together so that you're bringing something that can improve both welfare, safety, biosecurity, food production. These are all the areas where infectious disease can be really important. And I think for most of us, that's definitely been brought home in the last 12 months, really what an impact infectious disease can have on us as a society and an economy. As I mentioned at the start, the purpose of these symposia is to actually highlight some of the projects that we've funded throughout the last 3 years. In delivering the Bloomsbury Set programme, we've focused on some priming funds in different areas, and we've supported different types of project. We've supported longer-term project grants, individual innovation fellowships, commercialisation awards, the projects that you'll hear about today under the umbrella of AI and data science, and social science awards. So if we just focus on the projects we're hearing about today under the AI and big data umbrella, this programme of activities was delivered through 2 sandpits back in 2019, where we brought together researchers from our partner colleges and external organisations, really as a sandpit to brainstorm those big challenges, those big questions in this area where using data, AI or machine learning could really make a difference. During that sandpit, they worked together to develop ideas, which were then pitched to a panel of experts. Those experts then selected a short list of projects which were supported. 8 were initially in receipt of £2,000 to give them some start-off funds. And then after a second sandpit later in 2019, 5 projects were then awarded £30,000 to continue their work. We're going to hear 4 of those presentations today. If I can just move on to my next slide. This just provides you a little bit more input and insight into what the Bloomsbury Set is about, and some resources there where you can find out a little more information. There'll be recordings from today's event put up online, and there's also recordings available from our more recent vaccine symposium and our conference, which we host, and our first conference, which we hosted in February. And I'd just like to highlight that our second conference will be hosted in May. So let's move on and look at today's programme in a little more detail. I'm delighted to welcome our panel chair for these presentations and today's discussion, which is Dr. Gwen Knight, who's joining us from the London School of Hygiene and Tropical Medicine. Dr. Knight is an assistant professor who works on infectious disease modelling and antimicrobial resistance, and she'll be taking us through today's presentations. I'd like to hand over to Gwen. Thank you.
Thanks, Emma. It's my pleasure to chair this session of talks today. I'm really excited to see the range of both techniques and topics that we'll cover in these 5 talks. And I hope that we can have a great discussion at the end on how best to use big data to tackle these public health priorities. A final reminder to those of you watching live today to post any questions in the chat as we go along, and the time for questions will be at the very end. So I'd like to begin by introducing our first speaker. And this is Dr. Laura Merson. She's the Associate Director of the Infectious Diseases Data Observatory. Her research interests encompass issues around data sharing of biomedical research data, as well as how best enable rapid response to emerging infections. Her role as an executive member of the International Severe Acute Respiratory and Emerging Infections Consortium make her an ideal speaker to start us off during the current pandemic to talk about global data sharing. Over to you, Laura. I look forward to your talk.
Thank you very much, Gwen, and thanks to everybody for having me today. It's really nice to be amongst this group of experts and unfortunately as an outsider, but as an insider on the aims and objectives of the Bloomsbury set. And what I want to focus on is the importance of data and the importance of access to data, which has become all that more poignant over the last 12 months, I'm afraid. And so I'll give you a brief overview of our work towards accelerating pandemic response through global data sharing. And next slide, thanks. So it's widely acknowledged that sharing data on emerging infections is critical to an effective and efficient outbreak response. And this global recognition, plus recent technological advances, have led to some really outstanding examples of how data can be shared to accelerate response. And so I hope what many of you are familiar with is recent initiatives to publish viral sequences that enable vaccine development and help us to track the genetic variants of this pathogen and other pathogens, so we can understand the differences in how the disease presents and how it transmits. And also there's great initiatives in making global epidemiological data available to help governments prepare for spreading cases. However, access to clinical data has not kept pace with this trend at all. And why is that? We know that clinical data are absolutely critical to understand every aspect of a novel pathogen, especially in the early response. We need these clinical data to build case definitions so we can understand what this disease is and identify it. We also need to understand risk factors and transmission so we can build an effective public health response. And we need clinical data to understand the pathophysiology of the disease so we can target treatments, understand who to treat, when, and how. Next slide. And we also know that to understand the spectrum of disease, it's important to gather clinical data from a wide variety of people and places. Pathogens have a different effect on different subgroups of people, and each of which may only be present in small numbers in one area. So by combining these data, we can accelerate understanding of how this disease affects everyone, and especially in populations like pregnant women or children, or those with comorbidities who tend to be most at risk. Next. And we know that collecting and sharing clinical data is really hard. It's especially hard when all of the available resources are targeted towards an outbreak. And we know this because we failed. We, as the global research community, have repeatedly failed to mount an effective research response. In the 2003 SARS pandemic, the 2009 H1N1 global pandemic, all the H5N1 outbreaks that have happened in between, time and time again, we have not been able to respond within the time frame of an outbreak or an epidemic to be able to change the course of that epidemic. And so about 10 years ago, with 2 different hats on, I was a part of 2 initiatives that were looking to address this. Next slide, thanks. So what we decided is that in order to be able to respond effectively, we had to be prepared. In collaboration with the World Health Organization and ISARIC, we started to build a clinical characterization protocol with all of the case report forms and tools that you needed to execute it. So this is having the research framework there and available so that people can use it. In parallel to that, with the Infectious Diseases Data Observatory, we were assembling data sharing platforms. So this is the contracts and the governance and the systems in place, the infrastructure that you need to move data around and make it available. We wanted to assemble that and use that for other diseases such as malaria, visceral leishmaniasis, so that the infrastructure was in place and ready for the next outbreak. Then was the emergence of the SARS-CoV-2 pathogen. As soon as that pathogen emerged, we took those resources that we had already and the networks that existed around them, who were quite focused on infectious diseases and particularly severe acute respiratory illnesses. And we adapted those tools and all the infrastructure that we had for SARS-CoV-2 specifically, taking what we knew about that disease at the time and building it into the tools and the forms so that we could rapidly launch a response. Since then, we have iterated those forms with the new things that we've learned about the disease. When ACE inhibitors looked interesting, we started to add data collection on that. When a loss of taste and smell became relevant, we knew that we had to understand how prevalent this sign or symptom was. So we would add those to the case report forms and iterate the process as we went. And so I made these slides last week and already the numbers are out of date. So we've got almost 500,000 detailed inpatient hospital records of SARS-CoV-2 patients that are aggregated from use of these ISARIC tools onto a joint data sharing platform, and they have been made available for the global research community to use. On the next slide, you'll see that as of now, we've got, well approaching 500,000. It was 486,000 individuals last week from thousands of sites in 55 countries, so that we can look at the broad spectrum of this disease and gather novel evidence, especially on newly emerging variants, as quickly as possible, aggregate all of that data together and learn as fast as we can. But of course, all of this was only made possible by having the infrastructure and the governance to be able to share data collectively together. On the next slide, I'll show you a little bit about how it works. So these data are deposited to a platform by all the researchers around the world who collect them. If we have a collection of data sets from 2,100 sites around the world, we all know that those data are going to be in lots of different formats and very difficult to get together and use effectively. So we do that centrally. We curate all of these data to a single standard, which is called the CDISC standard, so that it's in a single repository with everything harmonized that makes it a lot easier to use. So researchers can come to our website and look at our inventory to explore the types and volumes of data that we have available and see if they could apply to their research question. If they do, they could write an application to the Data Access Committee. That committee reviews and hopefully approves or provides feedback on those applications. Those data are then made available under a data transfer agreement. They're of course pseudonymized, and we go through statistical disclosure processes to make sure that we protect patient privacy and enable release of those data sets so that researchers around the world can use them effectively to answer questions as fast as possible. So on the next slide, you'll see that we have 100% of the data sets that have been deposited to our repository thus far have been reused. They've been reused to date an average of 35 times because it's the breadth of the repository that really demonstrates how quickly we can accelerate evidence. So in lieu of using single data sets from single sites, what you can do is identify populations across the repository that can answer your questions. So we have very high levels of data reuse from all over the world. That last week was 17 countries. I'll have to increase those numbers by 3 more applications that we got this week. So it's rapidly building and rapidly supporting the infectious diseases community to address this pandemic and hopefully bring an end to it. So this is one example of how we can work together as a global scientific community to utilize data as effectively as possible for things that are happening real time and hopefully to save lives. Thanks so much for including me today.
Thank you, Laura. That was really interesting. As someone who actually has used the ISARIC data, I'm very grateful that you set this up before SARS-CoV-2 hit. So yes, I'll move now on to our second speaker, who's actually a colleague of mine at the London School of Hygiene and Tropical Medicine. So Dr. Michael Delves focuses his research on treatment for malaria, specifically on novel ways to block ongoing parasite transmission. He's recently been awarded an MRC Career Development Fellowship, and we'll be talking today about how he's using machine learning to tackle this important infectious disease problem. The floor is yours, Michael.
Thank you, Gwen, and thank you to the Bloomsbury Set for inviting me to share today. This is a project that is a collaboration between LSHTM, LSE and SOAS. And it's using, the aim is to use AI machine learning based image analysis routines to analyze drug-treated malaria parasites with the idea of accelerating anti-malarial drug discovery. Next slide, please. So I'm sure I don't need to introduce malaria in too much detail. It's a disease of devastating impact to both health and economic development of a country, and it disproportionately affects developing countries in the world. Just in 2019, even though we've made great progress, still 250 million people were affected by malaria, and there were over 400,000 deaths. So malaria is caused by a parasite called Plasmodium, and it's spread through the bite of mosquitoes. And transmission through mosquito bite is obviously important, not only for passing the disease around, but also for the spread of anti-malarial drug resistance, which is a real problem in the world at the moment. There is massive problems with frontline drug resistance in Southeast Asia, and just in the last week or so in Africa, the Lancet has reported that drug resistance has spread to Africa. And so whilst it's quite important to have drugs that prevent the disease in humans and cure people and prevent them from dying, also anti-malarial drugs that prevent this spread of malaria from humans to mosquitoes, so-called transmission blocking drugs, are highly sought after. But unfortunately, very few existing drugs have this property, and it's actually quite hard to discover new ones due to the complexities of growing the parasite in the laboratory and also high throughput screening. If you could move to the next slide, please. So my research for the last few years has been revolving around this question of looking for transmission blocking drugs. And I've performed high throughput screening of approximately 100,000 different chemical entities looking for transmission blocking molecules. And from that, I've identified approximately 120 anti-malarial compounds that prevent transmission of malaria from humans to mosquitoes in the laboratory, which is fantastic. But being a poorly funded researcher and not having enough research funds to study all 120, I really need to know how to prioritize my investigations to identify the best molecules that have the most chance of being developed finally into drugs. And in this process, trying to find ways of understanding more about these molecules, I noticed that whilst they all prevent transmission of malaria to the mosquito, they all do this in very different ways. And this can be visualized very visibly by using microscopy to observe the effects of the drugs on the parasite. So on the right panel here, these are malaria parasites that have been treated with 3 different drugs and allowed to simulate basically their infection with a mosquito. And as you can see, that these different drugs lead to very distinctive morphologies within the cells, or cellular phenotypes, as we call it. So the kind of the aim of this project is to take lots and lots of images of drug-treated parasites and try and get a computer to try and make sense of this. Can we identify what these drugs are doing? Can we categorize them and to understand and classify them to see which would be the most effective anti-malarial drugs? So the approach that we take is shown on the next slide. So the aim of this project was to take off-the-shelf machine learning solutions that have already been developed by other researchers rather than reinventing the wheel, and selecting those that are free to use and have very permissive software licenses that allow for us to reproduce them, modify them, and eventually potentially use in some kind of commercial application without any royalties or licensing problems. And so the pipeline that we developed was essentially taking these different modules and putting them together in a Python-based environment with four main steps. So the first was detecting the cells using these machine learning algorithms to identify individual cells from a large microscope image, pulling those out, and then extracting relevant features from this. So the routine that we use actually takes 1,000 different measurements from each different individual cell image. And then this is processed and clustered to provide some kind of meaningful classification of this, and which can then be visualized by the user using a web-based interface. And the whole philosophy behind the project was to develop a tool which was both extendable and flexible. So our case use here is malaria, but it could be applied to any set of images of cells. And we wanted something that was reproducible so that any researcher could take our data set, download it, run through the same analysis routine and get exactly the same result, and it would be auditable as well. And then finally, understanding that most biological scientists aren't complete computer wizards, have something that the average biological scientist can use, because existing tools to date are very, very complicated and not very user-friendly. And so the output from all of this analysis shown on the next slide. As I said before, it's a web-based interface which essentially plots all of the different cells within the analysis. And in this case, there were several thousand different individual cells with 50 different drug treatments, and plots them as a distribution, a clustered distribution. And so cells with very similar phenotypes cluster together, and cells with different phenotypes cluster in a different place. And this is an interactive process. So this web page can then be interacted with. It can be zoomed in on, as shown by the right panel. And the individual cells that make up each cluster, the images, can actually be visualized in situ. And they can be interrogated and looked at visibly, but also they can be clicked on to get the metadata of the image back. So where the image was located, what experiment it was from, what drug was treated, was treating these cells. And this information can all be gathered very quickly to rapidly analyze so which drugs form which clusters within your data set. And then finally, this information can then be exported for further customized analysis and looking in more detail, as shown on the next slide. So we're still probing this detail. This is sort of hot off the press, but one sort of simple analysis of the data that we performed is a hierarchical clustering of the different drug fingerprints. So the different phenotypes produced by each antimalarial drug. And actually, quite interestingly, what this data has shown is that there seems to be four sort of superclusters of phenotypes that are generated by all these different antimalarial drugs. And on the right-hand side, this graph kind of shows a simplified version of this. And these seem to represent, these clusters seem to represent the action that these different drugs are having on the malaria parasite during transmission. And so, for example, the green cluster here are cells that do not activate. These are cells where the drug has actually completely physically inhibited the process of transmission, whereas the other clusters, the drug has inhibited the process during transmission somewhere along the way. So this is actually super helpful for my research, as these drugs that do not activate the transmission pathways are going to be the best drugs for taking forward. So this is where already I can start to whittle down my many drugs that I want to study to a few key drugs to put most of my research efforts into. And I can now use this to generate an evidence-based hypothesis for their action and design my further kind of more detailed lab experiments to follow this up in more detail. So this has greatly accelerated my research and is a massively useful tool for me. In terms of future work to develop this, as shown on the next slide, so we're really pleased with how this has worked and how we've developed this in such a short space of time. But areas that we want to work with in the future is to continue to improve the AI detection of the individual cells within the raw image data. This works very well, but still needs more images to improve the model. Secondly, at the moment, this is a command line based interface, which is not so useful for a very a very novice computer user. And so we would like to develop a much more intuitive user interface, maybe sort of a web page interface or something that would allow more casual use of this tool by researchers to quickly get what they want and customize different parameters. And then finally, as I said before, this is a flexible tool that we believe can be used for many different applications in bioscience. And so we want to start to move away from just looking at malaria images, but also evaluate other data sets to show its utility at analyzing those. And probably the first pass of this will be to look at some of the large curated high throughput imaging data sources that are also already freely available on the web. So just to finish, I'd like to acknowledge my co-investigators for this award at LSC and SOAS, who've been an integral part to the whole process and provided invaluable advice and expertise. And a very big thanks to the software developers that actually took our slightly crazy idea and turned it into reality very, very quickly, headed up by Marco at MS Corp. And I'll be very happy to answer any questions later on. And thank you for listening.
Thank you, Michael. Very nice example of using big data to narrow down subsequent resources. It's really interesting. As Michael said, he's very happy to take questions. If you post them in the chat, then we can bring them all together after the panel discussion right at the end. So it's my pleasure now to welcome our third speaker, Professor David Broadbelt. He's a professor of evidence-based veterinary medicine at the Royal Veterinary College. As a co-project lead for VetCompass, an RVC-led project to collect clinical data from veterinary practice electronic health records, his research covers the use of big data for practice-based research. Today, he will talk about combining his research interests on companion animal epidemiology and this electronic health record data to address the problem of antimicrobial resistance. Very much looking forward to hearing your talk. Over to you, David.
Thank you, Gwen, and thank you the other speakers. So the moving species here, we're thinking about big data from small patients, and we'll be looking at companion animal related data. I'd just like to acknowledge my colleagues from the Royal Veterinary College, London School of Hygiene, and Manchester Metropolitan University. Next slide, please. So by way of introduction, antimicrobial resistance, obviously, we're all aware of it's a growing concern. It's also a growing concern in veterinary practice, and specifically researchers and public policy makers becoming aware it could be a growing concern in companion animal practice, dog and cat practice. There's certainly a link to prescribing levels, antimicrobials overall, and critically important antimicrobials in particular. And so monitoring levels of antimicrobial usage is important, and certainly there's ongoing work in animal practice, companion animal practice doing that. But also monitoring antimicrobial resistance via related culture sensitivity data would be valuable. Currently, those data are available, culture and sensitivity from private labs, and animal health records, certainly working here at the RVC within the project VetCompass Gwen referred to. But currently, the linkage of those two data sets is not available. Next slide, please. So our challenge here is then to try to link two de-identified data sets, VetCompass health records and laboratory culture and sensitivity records. The latter are returned to practices as PDFs, but because they're rich in owner and patient identifiers, we within VetCompass do not have access to them. Some of the results are recorded in the clinical records, but the vets only summarize these inconsistently. They may refer to file attached, spoke to IDEX, results now back, but the full records are rarely in the clinical notes. Hence, the value, potential huge value in linking two data sets to have a much richer data set. Next slide, please. So our challenge then here is to look first at VetCompass EPR electronic patient records that are de-identified. And here within the data set relating to dogs and cats, there are approximately 15 million patients and a related 500 million clinical and treatment notes. And so our first challenge was to identify the laboratory culture and sensitivity request within this rather big haystack. And then related to that, we had access to de-identified culture and sensitivity data from one of the big veterinary laboratories, IDEX, and here relating We had culture and sensitivity results from 330,000 reports spanning approximately 10 years, principally dogs and cats. And within that data set, in addition to the test results, we have practice postcode, lab request number, species, breed, and sex. But those were our only data fields. Next slide. So method then, first, working on the vet compass side, we worked to identify those culture and sensitivity submissions, and we used a rules-based approach to start with to find possible or potential culture and sensitivity records. And so from that, we found approximately 36, working within 2019, approximately 36,000 dogs and cats that had reference to plausible culture and sensitivities. We used that data set when we set loose 6 students to code manually 2,600 of those patients to establish whether they genuinely had culture sensitivity event. And that then powered a machine learning, a longitudinal note classifier, to classify at the note level, not the patient level, whether the note had evidence of culture sensitivity, classifying candidate possible cases and a random sample of notes were highly likely to be negatives. And then we evaluated note level performance. And the little picture there just highlights what some of our records look like. So we may have a range of treatment inputs, a number of notes, clinical notes, etc. And thinking about most patients having somewhere between 30 and 50 notes in their history. Next slide, please. Then on the IDEX side, we're looking to establish preliminary matches. And to do this, as a proof of concept, we used a rules-based approach from the Vet Compass culture and sensitivity events linking to IDEX. And so we had Vet Compass records with a submission date plus or minus 5 days around a specific IDEX test, and looking to initially link practice postcode and species on a specific date. We took a specific date, 19th of April, 2019. Next slide, please. And so in terms of our results, first of all, when we set up to clean the Vet Compass side of the data using our key search terms to establish our candidate cases, we use a number of terms that would be most likely to identify possible culture and sensitivity and individually establish their predictive values, positive predictive values. And then combined, when we use the set of key terms, predictive value of those terms is around the 80% mark. And this we found from our coded subset of approximately just over 2,000 of the coded patients were correctly identified as having a culture and sensitivity within their record. And this established then our 36,000 candidate case to then power the machine learning. Next slide, please. And so when we then applied to clean up the Vet Compass site to identify the culture and sensitivity, at the note level, we used the 2,600 coded patients as a training set to then run the machine learning on the test set and evaluated note performance, where the ROC curve was reasonably good, 0.99, or pretty good. And then the performance predictive value across a range of sensitivities was sort of generally good. This was at the note level, so approximately positive predictive value of somewhere between 0.4 or 40% down to about 20% across where we drew our sensitivity cutoff. Next slide, please. And this then allowed us to do our test matching of Vet Compass to IDEX data. And so on our specific date, 19th of April, there were 26 lab events that potentially matched to 131 Vet Compass IDs. And of those, using our rule-based, we identified 9 exact matches and 7 possible matches. Next slide. So in summary, the machine learning classifier for Vet Compass culture and sensitivity had moderate to good performance at the note level, but was excellent at the patient level. And it allowed us to clean the Vet Compass side for possible matching to IDEX from over 20-to-1 matches down to 5-to-1. And this then will set us going forward, one, to develop the matching on the lab side further, but equally the EMMA machine learning classifier that we used developed on the Vet Compass side can have applications identifying other terms, so specific conditions, other laboratory events, etc. And then future work also will be looking to extend our linkage to other potentially de-identified data sources, histopathology, other clinical pathological tests, other infectious agents, COVID-19, genetic and pedigree data. Last slide, please. And so I'll finish there and welcome questions later. And just would like to acknowledge again the RVC Vet Compass team, our collaborators, London School of Hygiene, Dr. Sam Clifford, and at Manchester Metropolitan University, Professor Yonghong Pan. Thank you.
Thank you, David. Again, very interesting talk. Nice to see how machine learning, again, is being used to link different data sources together in a consistent, structured framework. And great to be hearing about antimicrobial resistance. Thank you very much. Okay, so I'll move now to our fourth speaker, who's also from the London School of Hygiene and Tropical Medicine. And he has a research focus that makes him an ideal speaker for today's symposium. Dr. Jodie Fallon designs data analysis pipelines to process large data in order to provide biological insights into population structure, transmission, and drug resistance. His talk today will focus on genomics data curation and collection. Over to you, Jodie.
Hi, thank you. And thank you for the Bloomsbury set for the invite to speak. So I'm going to be talking today about a framework that we've developed that helps collect and curate genomics data. I'm going to be presenting this with a specific example looking at tuberculosis, but in theory can be extended towards any other organism. So on the next slide, I just highlighted some key figures regarding tuberculosis. So it still poses quite a large burden on global health services worldwide. It's about 10.4 million new cases and 1.7 million deaths annually. Resistance does occur to drugs we use to treat tuberculosis. And so about 500,000 annually patients are diagnosed with multi-drug resistant TB or MDR-TB. And importantly, only about 1 in 5 are actually enrolled on an appropriate regimen. So resistance is obviously a huge issue, and the accurate and timely detection of it is imperative. So we can detect resistance using a couple of different methods. So one of the first methods used is a culture-based method, where we kind of try to grow the bacteria in the presence of drug. If it grows, obviously it's resistant. The other method that we use is to look for mutations. And so we know that mutations cause resistance. And if we can characterize mutations on the genome using a whole genome sequencing, and so I put up here a slide looking at a potential pipeline that we could use. If we can characterize the DNA, we can then make a prediction on whether or not it's resistant. And so these sequencing machines produce a lot of raw data, and the volume of the data actually really makes a big data in a sense. That it's too large for kind of normal softwares to analyze. So softwares like R or Stata or even Excel can't really handle the type of data. And so what we've done is we've developed a program called TB Profiler, which runs on the command line and takes this raw data and a mutation database and provides a prediction of whether it's resistance or not. And obviously, this process relies heavily on the mutation database being complete, and our knowledge of the resistance mechanisms influences the predictive accuracy. And so on the next slide, I kind of shown you some of the studies that have come out, which look at trying to characterize these resistance mutations using what's called whole genome-wide association studies. These are a variety of different machine learning techniques, but in theory, they all use the same kind of inputs, which is your outcome. In this case, the outcome is the phenotype, which is being characterized by DST, and also the genotype, which is their genomics data. And so these two inputs are kind of crunched through, and your output is a list of important mutations which can provide a prediction on resistance and are quite informative. So this works well for drugs which we have a lot of data for, and we've characterized the resistance mechanisms quite nicely for a lot of the first line and second line drugs. However, there are some newer drugs that have come out And in the next slide, I kind of show three of the main ones, which have all been approved in the last decade. So delamanid, bedaquiline, and protomanid are new drugs, which unfortunately resistance has already been found in some clinical isolates. Resistance has been characterized in these isolates and they've been sequenced. And the mutations which confer resistance are actually quite rare, so they haven't been observed before. And this is expected. And hopefully, as we sequence more of these isolates, we'll actually see the same mutations coming up over and over. And so as we sequence and find these mutations, we can start to build up some kind of statistical support for them being predictive of resistance. And the idea is that as we add in more isolates, that we can see these mutations and select them for our database. So in the next slide, I'm going to show you this process. So here I'll just explain what this graph is showing you. Each point represents a single mutation, and they're plotted on the XY axis according to the genomic position at which they occur, and then the minus log 10 transformed p-value. So the higher on this y-axis they are, the more associated with the phenotype. And what I've done is, rather than to perform the analysis on the complete data, I've sequentially added in more and more isolates. And so you can see as we add more isolates, the support for particular mutations increases. And if we draw a certain kind of cutoff for this, then we can select mutations for our database. And so we thought about using this process to build some kind of a pipeline and framework for collecting data to inform resistance mutations for some of the newer drugs. And so on the next slide, I kind of show you how this would work. And so different users are connected to the database and they provide genomic data and DST data. This then, at certain intervals, gets put through a machine learning algorithm, which then produces a list of mutations which are informative for prediction. And at a certain cutoff, we can create a threshold at which any mutations which go past this threshold are selected in our database. And then this can be used by TB Profiler and other software. And so obviously one of the things that we need to do is incentivize users to provide data, which is always a bit tricky. So one of the things that we've tried to do is we've created a web-based platform for users to run TB Profiler. And so TB Profiler is actually a command line tool, so you need quite a lot of bioinformatic expertise to run. And so by providing a point and click interface, it makes it very easy for someone who doesn't have any knowledge to get a prediction. And so on the next slide, I'm going to show you what results they might see, including drug resistance, lineage prediction, and kind of predictions for the individual drugs on whether it should be sensitive, resistant. Further to that, on the next slide, I'm going to show you some other metrics that we show, which can only be calculated really if provided with DST data, including the power of the library to call resistance based on the DSTs, and obviously also just provide a timeline of the isolates that are coming in through the user, and predicting whether they're sensitive or resistant or not. Also, as part of that, we can use the data to actually identify mutations before they actually have any statistical support. So in the case of false negative samples, for example, we can look at genes which we know are causing resistance and identify particular mutations which may only be present in one sample. But by doing that and connecting with data from other users, we can then start to provide the user with some evidence or support, which they could potentially take into the lab and try to confirm. And so on the next slide, I'll just show you kind of the summary of what we've designed. So we've termed it TB Crowd, and really it's a crowdsourced detection of emerging resistance mutations. And so it's a system for sharing whole genome sequence and DST data. The users obviously get a dashboard with their aggregated results. And in the back end, that data is then used for the automated association of mutations. And though we focus mainly here on the framework for collecting the data, in theory, then lots of different algorithms could then be plugged in, and the most appropriate one could be selected. And we hope as resistance to new drugs increases, that TB crowd can then help find causal mutations in a real-time manner, rather than collecting it over years and then providing a result after maybe it's too late. So yeah, that's a really quick introduction to it. I'd like to thank everyone involved, obviously Bloom's Reset, and then myself, Gary Napier, a PhD student at London School, Ruby Chang and Martin Walker at RBC. And I'll be happy to take any questions later. Thank you.
Thank you, Jodie. It's really good to see focus on supporting clinical decision making as these new drugs are being used directly for TB. So trying to understand drug resistance as it's emerging and building up our evidence iteratively is great open source data that you're building there. Okay, move on now to our final speaker. Thank you to everyone for being on time, and a quick reminder to those listening in live that you can submit your questions through our chat function, and we'll bring them together at the end. So our final speaker is Dr. Martin Walker. He's a lecturer in epidemiology at the Royal Veterinary College. As a mathematical epidemiologist, Martin's research focuses on the control and elimination of tropical and zoonotic infectious diseases. He uses mathematical transmission and statistical models to assist with intervention and clinical trial design, and to identify atypical drug responses that could be a signal of emerging drug resistance. With a focus on neglected diseases, he'll talk here about how to safeguard treatment options. Over to you, Martin.
Thank you, Gwen, and good afternoon, everyone who is listening out there. So I'm going to talk to you today about our Bloom's Reset project that runs in collaboration with an IDO data sharing platform, data sharing and reuse platform, similar to the platform you heard a little bit about from Laura at the beginning of this session. And next slide, please. So you may or may not know, but approximately 2 billion people, mostly the most impoverished people on the planet, are infected with one of these beasts, schistosomes, or the so-called soil-transmitted helminths. So these are all parasitic worms, and they're amongst the most prevalent human parasites and so-called neglected tropical diseases. So schistosomiasis, very briefly, it causes abdominal pain, diarrhea, bloody stool, or blood in the urine. And those who've been infected for a long time may experience liver damage, kidney failure, infertility, or bladder cancer. And in children, it can cause poor growth and learning difficulties. Soil-transmitted helminthiasis similarly cause abdominal pain, diarrhea, anemia, malnourishment, growth retardation, and impaired cognitive development. Heavy infections can also lead to intestinal obstruction. Next slide, please. So in the last decade, there's been an unprecedented scale up to try and control and indeed eliminate these diseases. The main strategy is yearly mass drug administration, or also called preventive chemotherapy, of anti-parasitic medicines given to school children or entire communities. And in 2015, the WHO announced that for the first time, over 1 billion treatments had been distributed in that year. And this has continued ever since. So over 1 billion treatments a year, every year since 2015 and counting. However, the cornerstone of this strategy naturally relies on the efficacy of praziquantel for treating schistosomiasis and a couple of benzimidazoles, albendazole, mebendazole, for treating soil-transmitted helminthiasis. And yet, despite this massive use, we actually know fairly little about how these drugs are working in the field, and in particular, whether resistance might be developing. And resistance has been commonplace in similar drugs used in veterinary medicine. Next slide, please. So to counter this, we've teamed up with IDO to build a data sharing and reuse platform to assemble schistosomiasis and soil-transmitted helminthiasis clinical data. And this is a project that's been ongoing for the past few years. And the goal of this really is to spur and facilitate research on treatment responses for patients suffering from schistosomiasis and STHs, soil-transmitted helminthiasis. And next slide, please. So this slide shows you how our Bloomsbury set activities fit in with the IDO data platform development activities, and they're pretty much divided into two strands. The first is on the development of a study metadata and efficacy explorer, which we hope will enhance the findability of data hosted by the platform and also facilitate engagement and use with it. The second strand is on the development and application of model-based statistical methods to improve individual level inference on treatment responses. And I'll touch on each of these themes in turn. So next slide, please. So for this project, we've initially decided to focus on the STHs for the development of the Efficacy Explorer. And this map here shows where our target studies are in the world. So this is from a systematic review and scoping exercise, and indicates with the colors how many individual patient data we estimate are out there from different countries. So the darker the color, the more patient data. So this is not what we have, it's what we have scoped and what we believe is out there. So our challenge in developing the platform and indeed the Explorer is to get hold of these data. Next slide, please. So at the moment, we're still at a prototype stage for the metadata and efficacy explorer. So this is what it's looking like at the moment. It's an interactive map with the aim to show locations of the various different studies. And then one can browse, see where the studies are, click on the pins and get some information about the study. And also we want to put on there the estimates of efficacy. We'll also in time color code these pins to project a stronger visualization of how responses might vary between locations and through time. So ultimately we want to create something that's a little bit like an epidemiological map that some of you may be quite familiar with, but for drug efficacy. Next slide, please. So the other aspect of the project is on the development and application of model-based statistical methods to improve individual patient level inference on treatment responses. And essentially this is designed to provide new statistical methods so we can understand the distribution of individual responses in a population or community, rather than just being reliant on the average response, as has typically been the case for schistosomiasis and STHs. We can also use WHO criteria for what is considered a satisfactory, doubtful, and reduced response to treatment, and then summarize the number of individuals who are falling within each category. Next slide, please. So as a little illustrative example of this, these are some efficacy estimates for 3 STHs from a community in Pembrokeshire Island, Tanzania. And I'd like to acknowledge here our collaborators at the Starworms Consortium for contributing these data to the IDO platform. So on the top row, you can see the classical population average estimates of efficacy against the different STH species, Ascaris lumbricoides, Trichuris, Trichura, and hookworm species. And on the bottom row is the kind of information you get out from applying a more individual level analysis. So you can see that it gives you an idea of the distribution of responses that's behind that average, if you like. So if you look at, for example, hookworm in the far right-hand column, the average efficacy is around 84%, which would be classified as doubtful following the WHO criteria. But in reality, only about 12% of individuals respond in this manner. 68% are responding satisfactorily, and a further 20% have a reduced response. So ultimately, we want to include this type of information on our Efficacy Explorer, so we don't just get a single point population estimates of efficacy, but also some idea of the distribution of responses in different communities. Next slide, please. So just to finish, I want to thank the organisers for the opportunity to speak, funding from the Bloomsbury Set, and also Wellcome for the development of the data platform. and of course to all our many collaborators and contributors who've already submitted their data and shared it with us. Thank you.
Thank you, Martin. Very interesting to see the combination of the differences, the importance of the individual level data in understanding what's going on in terms of drug resistance. Okay, so we're going to open the floor up now to a panel discussion. So all 5 of our speakers will be joining in this discussion, which we hope will be around the use of big data to answer questions about infectious disease dynamics and antimicrobial resistance. So I'd like to start by again thanking all our speakers for their very interesting and insightful talks. Can I start by asking a question to everybody about the future? So about what we think big data is going to do within your research field. So potentially that's what you think the next big piece of data will be. What's the ideal data collection that you need in your field and how it's going to change it going forward? Since you are warmed up and ready to go, can I ask you, Martin, to carry on from your talk to give us an idea of what you think big data might do within your field and what the next big piece of data you think is coming?
Yeah, sure. Well, I mean, in terms of the work that we've been doing, I'm not sure I'd necessarily call it big data as such. Maybe medium-sized data, especially when you look at some of the data, the clinical data that Laura was describing, the sheer number of data they have for COVID. But I mean, the main barrier and the main thing that we come across is really, and this is possibly a more of a neglected tropical disease domain specific issue, is just encouraging buy-in and engagement with the whole idea of sharing data. There are enough data out there. There are plenty of data out there that could be made much more useful by assembling them together and having joint sort of individual based meta-analyses. It's just really banging that drum in the community and getting everyone comfortable with sharing data. So a lot of the time we spend doing that sort of communication exercises and convincing people that sharing is a good idea and is okay, and that will build up our mid-sized data, I would say.
Okay, so maybe I can pass that straight on to Jody then, because he touched on that a little bit in terms of getting the community to engage in the TB genomics side. So do you want to comment there, Jody?
Yeah, no, I completely agree with Martin. I think thus far has been quite a lot of, I guess, data hoarding. I guess it's always difficult in research and academia when quite often data is required for publication, and then publications are required for funding. But it is really trying to convince that the community that there is, I guess, an added value by the collation of data in a single resource, and that this will provide then again impact on clinical decision making. And maybe that's more that should be more kind of aimed at the clinical community. I know kind of in research communities, it's always going to be a bit of a delay between data generation and then uploading to a public resource for obvious reasons. But I think, yeah, I agree with Martin. I think that's something that really has to be worked on. But as for the future, I think, yeah, definitely as whole genome sequencing starts to become cheaper, I think there's going to be a lot of data generation, and it's going to be, I guess, very important to have the infrastructure in place to deal with all that.
Yeah, so that's great that you're setting it up now with the vision for more and more sequencing coming through. I guess we're already at the beginning of that huge amount of genomic data that's going to come through. Mm-hmm. Maybe David, you could comment there on we're talking a bit about the clinical side, but on the vet side, what I was wondering in your talk was how much what you're doing is being fed back to the note taking and what the veterinary practice is actually doing in terms of recording when they're taking sensitivity analyses and these kind of things, and whether you see data recording changing based on what you're doing, whether that's something going forward that you're hoping will happen for your research.
Yeah, it's a good question. And it's definitely a big challenge because certainly within that campus, for example, we work with about 1,500 practices, mostly within corporate groups. So we have direct communication with clinical directors at these big groups, but filtering down into sort of data recording behavior at the ground level is quite a challenge to sort of get things to be recorded differently, particularly just like GPs in practice, vets in practice have 15 minutes to do everything, 10 to 15 minutes, and they may not be that interested in what some guy at the RBC is asking them to do. So, so we have those challenges. I mean, there is the potential, and we have a separate project looking at trying to go forward, change behaviors of antimicrobial usage, for example, within practices, and so it's then working out how best to communicate and work with the vets who are actually in the clinics. Yeah, so it's definitely work starting out as opposed to even work in progress.
Interesting. And so maybe Laura could give some insight there on how she's managed to get very busy clinical individuals to fill in these ISLRIC reports. That seems like something that has been really well done during COVID, but seems to have also gone through ups and downs, which obviously for the data user is tricky to know, like what's the denominator. That's always the problem, right? So.
You're right about that, and we're at the mercy of the pandemic status, if you will, in any one hospital that we're managing. But I think the incentive for the physicians who work with us is that they're seeing immediate impact. So the data that we collect, we produce what was weekly and is now turned to monthly reports. So there's an immediate feedback of the aggregate international data to everyone who's inputting data onto the system. So I think the most important thing to get people engaged is do something with the data that's relevant to them and what they're doing. And I think to do that better in the future, something I would like to focus on is data standards. Very simply, for example, around definitions. So what is suspect COVID? And how do you classify that? And it's incredibly difficult to compare the numbers that you get across a number of different sites if we don't have uniform definitions, and even better, uniform ways of collecting and standardizing, harmonizing data. Then lastly, I think what we really need to work on is linking data better across disciplines. So to have the genomic data interoperable with the clinical data, to bring in the epidemiological data, all the other omics, I mean, that I think is our next big challenge that can turn clinical data into big data, not just by numbers, but by working in a cross-disciplinary manner.
Yeah, I can see Jody nodding. I don't know if you want to come in on that, on the integration of genomics with other data.
Yeah, no, I completely agree. I guess it's just the volume of genomic data makes it very difficult so far to have been included with other data, which can generally kind of form or fit into kind of a square table form. I guess the unstructured nature of genomic data, just makes it difficult. But I think, especially probably driven by the pandemic and the importance of sequence data, we're probably going to see a bit of a revolution in terms of how to integrate that into other databases.
Because I guess that's something that I've been thinking about in terms of, for drug resistance, we have very clear definitions, right? And we have bodies that regulate those and define phenotypically what is resistance and not. But then I guess what we heard from Michael was more about other ways that we can think about resistance. So there's some more like the visualization of resistance, which again, some people have been working on in TV, right? Thinking about how can we watch bugs grow and use that as an indicator of resistance. But Michael, I think you're talking more about actual cell shapes and how that varies and things. So I don't know if you want to comment there about how much your work, you're not really relying on other people to input data, you're more building a structure up for them to use, and then that becomes very important that you have standardization or cross data linkage, I guess.
Yeah, for sure. So I mean, for me, as you say, data sharing is not so much of an issue, but the data generation itself is the bottleneck. Needing to image hundreds, thousands of different samples requires complicated or sort of automation, automated microscopes and data experimental setups and things. And whilst it is possible to standardize these things between different labs, there are kind of quite a lot of challenges with just getting images that on one microscope, even in the same room as another microscope, looking similar. So having others contribute to the data set is fantastic, but it is quite problematic. And I think there are solutions there, but they still need to be developed and harmonized. I'm quite fortunate working in malaria that there is very excellent collaborations between academia and industry, facilitated by various organizations such as the Medicines for Malaria Venture. So access to expertise in these areas is really, really good. And so all of my experiences of data sharing and protocol sharing has been fantastic, really. So I'd highly recommend collaboration.
Yeah, I guess that's something that we had quite a nice mix, I feel, of people using existing technology and then sort of bringing that together into their own ways. I guess Jodie and Michael in particular, I suppose, there. But I wonder, Nora, whether you have a perspective on that. Is that something that we need to try and limit, like people coming up with their new framework for analysing data and learning the lessons as they go? And how can we better share as a community ways of working on such complex integrated data?
Well, I think what's important is that every bit of knowledge builds on the last. And so what's critical for that to happen is discoverability of that, well, not just of that paper that reports on the results, but ideally of that code and the data that underpin them. And so I think that ensuring that we've got machine readable, findable data that people can easily access and hopefully utilize, that they can not have to repeat anything. That would be our best way to ensure that we're accelerating science as fast as possible.
Yeah, I think that is so key. Not only for as you say, from a paper, the reproducibility of that paper, but then the ability to use it on your data set going forward. And I think that's where potentially, as Michael just said, collaboration with the private sector or cross-disciplinary working, right? Like as a microbiologist, you want to use these tools, but you couldn't code them yourself. So how do we foster cross-disciplinary conversations? I don't know if anybody wants to step in there to talk about what they've learned about the limits of their own abilities and how working with other people has helped them to enhance the use of big data. Martin, you had a brief nod there. I don't know, did you come up with all of those big Google, well, they look like Google Maps, sorry, those big maps yourselves with the coding of the pins and everything, or is that existing code?
No, that is stuff that we've done ourselves. Yeah, I mean, perhaps that's not the most efficient way of doing it. There are companies and things that specialize in that type of thing. So, I mean, perhaps, yeah, another approach would have been to try and outsource that part of it. It's difficult, I suppose, at the initial stages where you're exploring how you want things to look and be, and you're developing them. You want to keep a lot of that to yourself, I think. At a later stage, I suppose, when you're clearer on what you want your outcome to be, then I think there probably are more efficient ways of using private companies and things to do that.
David, I think you wanted to step in there, did you?
Yeah, I think, well, within our collaboration, there were definitely parts that, as on the epidemiology side, had very little understanding of what they were doing. So we definitely needed the collaboration across disciplines, to link the clinical to the machine learning and such. So, yeah.
And so how did you establish those links?
Well, this was through the Bloomsbury set. So it was through the sandpit. I mean, obviously we have our own data scientists, but it was building the links with colleagues at London School of Hygiene and elsewhere for getting the sort of full picture. Yeah.
Okay, I think we've had a few questions come through on the chat, so maybe I'll move on to asking those. So, Michael, you've had a question about whether you can use your work for schistosomiasis. So a really interesting talk. I heard of similar initiatives to identify potentially effective treatments for schistosomiasis. Are your methods similar to this? Could they use the same platform?
Yeah, so I'm not familiar with that particular study, but the way that our pipeline has been developed, it is sort of purpose agnostic. So as long as you can provide images of the cells that are in great enough volume with metadata provided, then yes, that would work probably very well. At the moment, so we're using without telling you technical, fluorescence microscopy images, which simplify things because you highlight only certain parts of the cell in different colors. So in this case, we're actually only using 2 different colors, but there's no reason why you couldn't use many more colors to measure many more parameters, or even sort of full color images, such as slides from a patient or something. As long as you can image it in enough quantity and provide some kind of training set or annotation to some cells, then in theory we can tailor the pipeline to give some kind of output that's meaningful for the person.
Great. So that's really important to know what it is that you need as an input and to generalize that as broad as possible, right?
Yeah, I mean, our kind of long-term goal is something that someone with not so much specialist knowledge could understand and use intuitively without having to know all the complicated computer code or anything. Maybe just through selecting various parameters and in menus and such, like get an answer as quick as possible back, basically, because I think most scientists aren't so interested in what's going on under the hood of the software, but getting the answer as quickly and as efficiently as possible, and being able to then validate and verify it.
Mm-hmm. Interesting. So then we have a question for Laura. So please, can you say a little bit more about how one can contribute to the IDDO and how to access data on your platform?
Sure. Well, the easy answer is check out our website at iddo.org. We run a number of different platforms, which Martin put up a slide that showed the different diseases that we cover. So within each of those, for the platforms that are active, there's an option to contribute data. We take data in any format, but usually with a focus. So with COVID, we are focused on data for clinical trials or inpatient hospitalizations. And so if you have any clinical data that relate to that, provided you have the appropriate permissions to contribute it, you can upload it in, again, any format directly to the website, and we'll curate it to the platform and take care of all the governance with you. Same thing to access data is the other selection on each of those platforms. Each of our platforms has a data access committee, so it always involves an application to that committee and making sure that you meet the requirements for access to those data, and of course, executing a data transfer agreement. But our aim is to maximize the reuse of data. So we very much want to support you to have access to data, to ensure that we do that in a very robust and secure way, but to facilitate it as much as possible. So please take a look at the website, select your disease, and just look to see what's available on inventories for those.
That's great, thank you. So sort of building a little bit on that, I guess, could you comment on the model for the long-term sustainability? So we've had a question that's probably relevant to all of us here, in that the platforms that we're developing require constant updating in real time, not just for the data, but also as the new technology comes through. Is there anything we need to learn or we need to take care of for ensuring long-term sustainability of the platforms that we're developing and the data access that we're sharing? When we share into a big data set, how can we ensure that we can continually access it going forward?
Yeah, so I think that DOIs, or persistent identifiers, are really important for being able to locate those data going into the future. But as we all know, software is going to change, technology is going to change, and future proofing against that is difficult. One way to do that, of course, is to not only save your data set somewhere where people can find it and access it, but to also contribute it to a repository that will take responsibility for curating it and protecting it. And that's what we've committed to at IDDO, is to bring the data sets into a larger repository, which is sustainable only because we have so many different diseases and therefore funders who continue to be interested in supporting our work, but can have a long-term commitment from another organization to secure those data and make sure that they're available in years to come. It's especially important with neglected tropical diseases or with emerging infections that have very small populations sometimes that are affected, or very disparate, geographically or temporally disparate populations. And so if you want to build a knowledge base on Ebola, the 90% of the available data was from the West African outbreak, which all but dissolved after that outbreak because the NGOs left and the ministries moved on. So that's one of the platforms that we have taken on to secure and protect those data to make sure that they're available in the longer term.
Oh, that's great to hear. And it's interesting to hear that the diversification away from a single disease has led to the long-term sustainability. So I guess then to ask Jodie, who's got more of like a long-term TB focused platform, and then Michael, who's just starting out with this kind of platform development. Michael, are you already thinking about long-term sustainability? And Jodie, what are you doing to think about long-term sustainability? Because your kind of platform's been around for 4 or 5 years now, right? Is that from one long-term sustainable funding?
Yeah. So I think so Laura's kind of commented on data, and so I'll maybe choose the other side of the platform, which is the actual software and the code that runs and creates the platform. And I think it's quite important for the sustainability of the platform to make that code available, to open source it, if obviously only in certain circumstances where there's no licensing issues. But that does mean that, for example, if a code maintainer for any reason can no longer fulfill that role, that someone else can then take that code and maintain it, and it's more of a community-based effort rather than a specific organization, and that's maybe more important for platforms like myself and maybe Michael's, where it's I guess right now having a smaller user base than IDDO must have. But yeah, I think to make the code available is also quite important for the sustainability.
Yeah, I would completely agree with Jodie there. So rather than reinventing the wheel, I mean, there are, for microscopy, there are already open data formats you can use, an open data microscopy repository, so you can put your data in. So I think probably as we generate more data, we would try and deposit it all in there so that you can it can be discoverable and linkable, persistently linkable, but also the code itself. So we're building upon freely available code and packaging up ourselves. At the moment, all our code is hosted on GitHub, so it can be accessed and downloaded by users, and eventually we'll be planning to sort of make that available. And if we explore some commercial angle in the future, that will be around the actual models used to identify the different cells and from the different applications that we're looking towards. So yeah, no, I think having an open approach, especially when you have quite niche people who are interested, it's important to be able to support the small community and yeah, help people along really, because I think through aggregating the data and other people contributing, you get much bigger impact in the long run, and it's the cross-pollination even of different people's work and different people's ideas are where all the interesting things for the future generally come from.
Gwen, can I just make a comment about this idea of open data? Because I think we're really witnessing a paradigm shift away from, let's say, classical academic culture, which used to be quite conservative with respect to having data or other such things available. And it's nice to see that our generation, I hope, is going to be leading that. And the reason why I don't think that we should be afraid of it is because increasingly funders and academic institutions are understanding the importance of it and the importance for accelerating science. And furthermore, are learning to acknowledge it and accredit it. And so take that into account on the things that count to us, which is their funding applications or academic appointments, et cetera. And so there's initiatives such as Make Data Count that are really learning how to assign DOIs to things, assign ORCID IDs, which identify the people involved in science, and then link those to the credit taxonomy, which tells what role you had in that science, and link all of that data together very robustly so that in the future, someone can look you up and see not only the paper that you published, but also the code that you made available, the data set that you shared, and all the different ways that you've supported science to move forward faster.
Yeah, I think that, as you say, that's happening right now and has only been accelerated by the current pandemic. Even over the last 5 to 6 years, as a mathematical modeler writing code for a lot of my papers, the shift has dramatically been to making that open access and readable, not just in some random repository, but making sure that has long-term sustainability and usability. And we've had a question about the sharing of human and animal data, which I guess is relevant both to David and Laura. Laura, I assume that the IDDO is not specifically just human clinical data, right? But there's obviously a need to make sure that we're thinking, especially in this time of One Health and zoonotic diseases, and of course antimicrobial resistance, how we can compare and contrast and just know what data there is about diseases with animal or human hosts in the same geographical settings.
Very much so, and I think that leads into my comment around cross-disciplinary approach. I think a One Health approach is absolutely critical, especially in the realm of emerging infections. That is something that I put on my list for what we need to do better, and not necessarily what we're doing well right now. IDDO does focus on clinical data, and the reason we do that is because, in my opinion, you need to build a repository around the scientific questions that you can answer. And those that we have selected are focused on treatment efficacy and patient-related issues. But I think that we as a community need to define other critical questions that can equally be brought forward as we make our data more interoperable and shared. Over to David for that comment.
Yeah, well, I mean, I was thinking more, I think when your original question was around how we're aware of the data too. No?
Yeah. Which goes to something Martin said, right? I thought was interesting about trying to scope what's out there and then try and find it, kind of thing.
Yeah, and certainly that on the animal side, I guess it's very species specific, probably strongest on the farm and production side, with less traditionally, well, that's changing, but traditionally less on areas we're interested in companion animal. But it is, I would just repeat, as Martin, it's a challenge to know what is there, and the first exercise is to find out what is there, both nationally, internationally, etc.
I'm just looking at a question. I just want to follow up with Laura on that idea of clinical, of what is your question, because I think I always have a concern with big data that we collect it for the sake of collecting it, right? And we need to make sure that we're collecting it and being aware of the limitations as we collect it, and then what answers can be answered with that big data. I don't know if that's something that's in the way that IDDO is structured, or is that, it sounds like it is from your question. I just wondered if you had a comment about that.
Yeah, what I can comment on is building repositories. So to have a repository, the governance structure that's around it, the infrastructure, and especially what we do, which is just, of course, curating data to that harmonized format so that it can be used more efficiently, it is incredibly resource intensive. And so we cannot do that for all data. The way to make that efficient is to focus on the questions that we really want to answer. Our group was founded on questions around anti-malarial resistance, and the idea that we had to bring together the global data in order to be able to address questions around the clinical resistance in antimalarials. I can let Martin comment about our work on schistosomiasis, of course, around COVID and emerging infections. We really focus on clinical characterization of disease. So what is this disease? How does it work? Who's impacted? And especially subgroup populations. Now that we have that better figured out, we move on to treatment efficacy. And how do we aggregate all of the clinical trial data to understand what's really working across different populations with all these tiny little studies that are being done.
It's the harmonization that's important. Martin, I don't know if you wanted to comment on that.
Yeah, so I mean, there's a slight chicken and egg balance here, because when we start these repositories, we need a certain amount of data to be able to do anything. And so the initial stage does often rely on the community. So in the example that I work in, the NTD community be willing to, sort of a core of them, be willing to share data and believe in the idea. But then to go beyond that, because that's a little bit like a standard collaboration amongst scientists. You can get relatively large groups of scientists who will share their data with each other. But to go beyond that and try and make it a truly inclusive and global platform, we do now think that we need to set out the research questions almost beforehand. So we know that certain data are out there, we know what the interesting questions are and what could be answered. And then we encourage contributions and engagement focused around that research question to try and increase the buy-in and the engagement with data sharing. Because I think there's sort of just share it because people are happy to share on their goodwill. It will only go so far. contexts are different. I mean, NTDs is a specific context, and with acute emergencies like with COVID, perhaps people are more generally engaged in that process already. But yeah, we've certainly found that now centering the research questions is an important way to encourage contribution.
Yeah, so we've had another question, which I guess goes to that. Rather than thinking about what the research questions are for the data, or should we collect the data and then work out the questions? We've had a question which is directed to Laura, but I think maybe we could all answer it, which is about basically what the collection of data does to reveal what the gaps are. So the question was about what can we do to correct gender imbalances? So the idea that there's a low representation of women in clinical trial data for therapies for NTDs. And is this a big problem? But I guess if anybody wants to chip in about how collecting big data or how trying to collect data has revealed the gaps that there are, and potentially that directed for you, Laura, was about the gender imbalance for clinical trial therapy for NTDs.
I can comment briefly and then hand over. Again, I think Martin knows this better than I, but I think the first thing to do is to identify imbalances. Mm-hmm. And you can only do that when you have more than one source of data. And so again, the strength in the evidence that you're producing is only as good as your sources of data. So in collecting the global data on visceral leishmaniasis, we've recently put out a paper that shows the disparity in that disease between boys and girls who are infected. Mm-hmm. And so only by putting together all of the different clinical trials that exist, did we have sufficient power to be able to demonstrate that. So I would say that is definitely one of the things that the power of big data can do, or bigger data.
Martin, did you want to come in on that?
Yeah, just to add, I mean, this is partly the reason that we do these fairly in-depth scoping exercises beforehand. So we look at and get what information we can from what's been published. That's obviously not the raw individual level data, but we do that to get a feeling of what is out there and what questions could be answered. So for instance, in the gender inequality, that we would often be able to get some sort of estimate as to how many boys and girls, men and women were enrolled in a trial from the paper. So it gives us an idea of really what the ultimate usefulness of the platform can be. And similarly, you may do a scoping exercise in a disease and find there just aren't that many data out there, and in which case you need to know that, because you don't want to start developing a platform when there aren't many data. So yeah, I mean, the scoping exercise is very important at the beginning of this.
Jodie, did you unmute specifically to chip in there, or? No?
Yes, I did. Yeah, just a quick mention also on that issue, that obviously sequencing is quite expensive. So most of the countries outputting sequence data at the moment are quite wealthy, and the strains which are present in those countries might be different from strains present in other countries, and that also might lead to different mutations being present in those strains, and that the actual the predictability of the resource being different because of that. So it's something that we should definitely look into.
Whether there's any barrier to including your data, because MDR, I think I dropped out of it. MDR TB data can be politically and economically sensitive. So I'm taking Laura's question here because that's in front of me. I hope that's okay, Laura. So do users of your platform need to enter information about who they are and where they work? And could this potentially be a barrier to uptake of the platform?
Yeah, so the idea is that there will be minimal amount of data collected. I guess there's three levels. There's pathogen data, which is the actual genomics and the DSTs. Then there is data on the submitter. So usually that would be a researcher or maybe someone who works in the clinical field. And then there's actual patient level data. So at the moment, we're not collecting any patient level data. So it's really on the pathogen and minimal information from the user. And that's mostly just to kind of, I guess, ensure some kind of accountability for the data, and also maintain some integrity of it. So if someone I guess minimally we want something like where they're submitting from and what field they work in, and potentially the kind of setting they're collecting from. But at the moment we're leaving it to something quite basic to not discourage people from providing too much and not wanting to submit data.
Jody, if I may, I think you've really highlighted 3 different layers of challenges to access to data there. There's the genomic data, to which there's an infrastructure barrier, which makes it very difficult for people who don't have high levels of resources to interact with or be a part of and set up. Then the patient level data, which of course has lots of ethics approvals and challenges around privacy, but then also the institutional level data in countries where reporting high consequence pathogens is very heavily regulated by the government. And I've worked in some of those countries as well. So I think it's a good way to illustrate all of the challenges that we're all facing at many different levels of being able to do what we want to do, which is treat patients better and save.
And I think that's a great place to end our discussion today. Thank you very much to all the speakers and all the interesting conversations we've had there. Pass over now to Emma, who is going to wrap up this session.
Perfect. Thank you, Gwen. So it's really great today to hear insight into the range of ways that data is being used in this space and where some of those future challenges and opportunities are. So thank you again to all of our participants today and to our audience who've asked some really great questions. Apologies to those that we just didn't have time for today. I'd also like to thank Gwen for being such a fantastic chair. A recording of today's event will be available on the website very soon for those of you who would like to watch it back or if you are unable to join us at the beginning. I'd just like finally on our last slide to highlight our next event, which is our conference Connecting Capability in Knowledge Exchange, which is taking place between the 25th and 27th of May. Those of you that have joined us live will receive an email with detail on how to register for that. And we do hope that you join us for the conference or for a future Bloomsbury Set event. So thank you again to everyone for your time today. Goodbye and enjoy the rest of your afternoon.